And now for termination. Okay, that's a dumb joke. All right, let's get our lecture back up and going there because I don't want to leave you guys hanging here. All right, termination. So there are three types of termination in eukaryotic systems. Remember, we're dealing with things now that have a nucleus. So there are different types of RNA. There's your transfer RNAs that carry around amino acids in the cytoplasm. Okay, so the transfer RNAs are going to be made in the nucleus, right? So I'm just going to make the nucleus really big. And they have to be transported out through what? Nuclear pores. Nuclear pores are the nuclear doors in and out of the nucleus. So transfer RNAs and ribosomal RNAs have specific sequences that initiate, that allow termination to happen that are different from messenger RNA. There are two models, really, that termination occurs in eukaryotic cells. So one model is called the allosteric model, which you see right here. Let me get another color. The allosteric model is where the RNA is cut near what we call the poly-A signal sequence. Now, what is that? Remember at the end of bacterial or prokaryotic messenger RNAs that you had all those U's? Well, here you have lots and lots of A. Okay? So, as I mentioned before, you know, this poly-A or polyadenylation signal or sequence can be very long and very much like the U's. Remember, A's and T's, very unstable, a lot less stable than G's and C's that have 3 hydrogen bonds, okay? So, the allosteric model, in this model, the RNA is literally cut, okay, near that sequence. So basically you have an enzyme that comes along and cuts it off. And so that is one way that this can happen. The other way is the torpedo model. Yay, torpedo. I don't know, torpedo model, right? The torpedo model states that the messenger RNA is cut near the poly-A signal, but the RNA polymerase continues for a little while. So what's going on here? So over here in the torpedo model, here is your polyadenylation sequence, which you are going to find here in the actual gene. It's at the end of the gene. The poly-A sequence is going to be basically a recognition site for enzymes to come and cut the terminal end of the messenger RNA off. The RNA polymerase continues to synthesize new RNA, okay? So in the allosteric model here's allosteric. Let me get my highlighter going here. The allosteric model over here is showing you that after the ribosome, not the ribosome, the RNA polymerase, sorry about that, passes that poly-A signal, RNA polymerase 2 becomes unstabilized, and basically enzymatic action cleaves this messenger RNA at the 3' end of the poly-A signal. Over here with the torpedo model, okay, there is what we call an exonuclease. Exonucleases are enzymes, all right? This is an enzyme that's going to hop on after that polyadenylation signal and start cutting up what's left of that little fragment of messenger RNA. Those little pieces of messenger RNA, remember, all of this, both models, let's just cut this in half, would be happening in the nucleus. So these little RNA fragments, are floating around in the nucleus, and they can be recycled, basically. There are enzymes that'll take these little fragments and break them up into individual nucleotides, recharge them for the production of new messenger RNA. So what happens to the RNA after it's made? Well, in eukaryotic cells, you have RNA modification. So, and it turns out even in prokaryotic cells, ribosomal RNA is modified. Transfer RNAs and ribosomal RNAs are modified by specific enzymes so that they have the appropriate shapes to do their jobs. Messenger RNA has to have additional modification to do a couple different things. Number one, there are two processes called capping and polyadenylation, which means more A's added, okay? We had that little tail chopped off. The tail's going to be added back on. So let's look at, and I know I have a picture of this, but we might as well draw it out just for our own purposes. Okay, here's the poly A tail. This is going to be 3'. The cap, this is a really interesting phenomenon. Actually, this is one of my favorite things about messenger RNA. You have a 5' cap with a methyl group on it. So basically, you'll have an MET here, all right? How does this happen? Basically, one of the places where you see, well, the place that you see ribosomal RNA being processed is in the nucleolus. All right, remember what the nucleolus is, you guys? When you have the nucleus inside of the cell, the nucleolus is the dark kind of ball structure that you see in all of the chromatin floating around in the nucleus. That nucleolus is where ribosomal RNA is being transcribed and being cut up into the ribosomal subunits. The subunits are prepared to exit then out into the cytoplasm where they fold up into their ultimate structure. So ribosomal RNAs are initially transcribed as one really long piece of RNA, okay? That would be the small, that would be the large. And basically what has to happen is the replacement of these little the excision of these segments here so that you can liberate the small fragment from the large fragment. And in fact, there you go, a much nicer picture, right? Sometimes I still like drawing it out. So this is what we call the 18S or 18 small, 5.8 small, 28 small. These can be found, don't worry about why we have different sizes right now, that's a different deal. But basically, here is the DNA. It's transcribed into ribosomal RNA. Each one of these fragments is removed in between these pieces of ribosomal RNA, and the 18S, 5.8S, and 28S, our RNA fragments are liberated and they can be transported out into the cytoplasm. Transfer RNA is a little bit different. So basically what happens with transfer RNA, and this is pretty consistent between eukaryotes and prokaryotes, is that these transfer RNAs have really, really big, what we call precursor RNAs. In other words, they're large pieces of RNA that get cut up into multiple transfer RNAs. And some of them are modified. So in this particular case, what you'll see is a large piece of transfer RNA, and then exonucleases and endonucleases are going to cut at these points to liberate the transfer RNA. This site here, the ACC, is where the amino acid attachment site is going to exist. And over here, this is basically like the 3' end, all right? We have a few covalently bound